So the last part of this is about viruses. So viruses are sometimes called non-living. And this is because they don't make their own energy. They cannot live all on their own. They don't have any organelles. And they can't survive without a host cell. Everything from the common cold, like the coronaviruses. So coronavirus as a virus-like group, it just causes a cold. In fact, a lot of people that had COVID, they just had a bad cold. Like they just had a lot of like snotty nose, some low-grade fever, but it wasn't terrible. Flu, the influenza virus, which is like having a heyday this year. Like there's a lot of that going around. Everything from just the cold types of things to like warts, herpes, which is where you get sores, like cold sores. Hepatitis can end up causing liver damage. Mumps, mononucleosis. All of these examples of viral infections, some of them lead to just something that's mild. Some of them can lead to very serious diseases. All viruses, though, have a couple of things. So one, they got some kind of nucleic acid inside. They can have DNA or RNA, but there is something inside, and that's pretty much it. There's DNA or RNA, viral DNA or RNA inside, and it's got a cover. The cover is called the capsid. So the cover really just protects the DNA or RNA. Then, so we have the capsid, and then there's an envelope, and the envelope is like an outer cover. The envelope is what helps it to stick to stuff. The envelope oftentimes has proteins on the surface that helps it to bind to certain host cells. So these bind to host cell surface. Okay, so you got genetic information with the cover, and then you got this outer cover that helps it to stick to things. So here's what happens. So you have what they call a host cell, and this could be any kind of cell. It could be a cell in your nasal passages. It could be a cell lining your GI tract. It could be cells that are on the skin surfaces. It's a host cell. The virus binds to it, has to be able to attach. Once it does that, then it actually injects its nucleic acid into the host cell. The nucleic acid of the virus tells the host cell, make copies. Make lots of copies of the nucleic acid, make lots of copies of the capsid, make lots of copies of the envelope or the cover, and package it all together. We are talking millions. So one virus particle infects a host cell and tells that host cell, make millions of virus particles. Once those virus particles are all packaged together, they get released. Now they can infect 1 million cells. And they can all tell those cells, make 1 million more virus particles. Do you see how this like rapidly increases in number? You go from one virus to 1 million, and then 1 million times 1 million, and then 1 million times 1 million more, right? So it just starts to increase and increase. To try and block this, we use vaccines. So vaccines are oftentimes a weakened form of a virus. So they take a known virus and they somehow change it. So they used to just boil it. They're like, well, we'll just boil it, it denatures, and then we'll use that and inject that into you. It's denatured, it's damaged, it can't cause disease, but it will trigger an immune response. That's really what a vaccine is for. Vaccines, you want to stimulate the immune system of the host so that they make antibodies. Because remember, antibodies stick to foreign substances. So antibodies then can help to bind to any foreign virus or infected cells and help to destroy them. So they don't have this ability to churn out millions and millions and millions of virus particles. So there's sort of like the picture. Virus infects, takes over cells, makes millions of virus particles, and now they get released. So retroviruses are a special kind of virus. Retroviruses, instead of having DNA, they have RNA. So this is sort of a challenge because what do we know about RNA? How many strands does RNA have? 1. Remember that it's a single strand? So if I am a retrovirus, then that means that I have AUCCGAUU, right? So I've got just a single strand. When you go to copy this, there's no pattern to compare it to. So DNA, remember I told you if DNA has a mismatch, it ends up making a bump in the strand. So like if you match AA and a mistake, you have repair enzymes to fix that. There's no two strands to compare. So retroviruses, the enzymes that copy retroviruses, they make mistakes a lot. They make a lot of errors. So you can see over the course of viruses infecting, replicating, infecting, replicating, infecting, replicating, you can have this one strand can end up being changed. And not just one time, but you can see then another and another and another. So every time that virus goes through replication, the odds of a mistake go up. This means that retroviruses mutate at a higher rate. This makes it difficult for your immune system to figure out the virus. Because if your virus has this sequence and it makes certain proteins and infects the cell in a certain way, your immune system can detect that, figure it out, and make antibodies to block it. But then what happens when there's a mutation and that causes a change in the virus? It might cause a change in the surface of the virus. It might cause a change in how the virus infects. And that means your immune system has to figure it all out again. So there's two viruses that are retroviruses, and one of them is HIV. So HIV stands for the human immunodeficiency virus. So this virus first came out kind of into the news and everything, started talking about it in the late 60s. Initially, they said, oh, well, this is seen only with homosexual men, and it's much more limited. They don't really know what it was, but they just saw that these people had this immune collapse. Their immune system, they had no immune response to things. They got really strange infections. They ended up with cancers that most people don't get. A healthy immune system would actually attack and figure those out and remove them. They would never develop. But here, HIV, this virus actually attacks and infects your immune cells. Specifically, one of your T cells. So T cells are a form of B cell, they're a form of lymphocyte. They infect immune cells, infect the cell, replicate, destroy the cell. Then all those virus particles go out and they infect more immune cells, replicate, and destroy those. So what happens to your immune system? Yeah, it fails. So now, if it infects the immune cells, you have no immunity. They are immunocompromised. Which means they have a weak immune system. And they end up with infections that they can't clear. So if they get a cold, it becomes bronchitis. It becomes pneumonia. So it doesn't just stay mild. It just keeps getting worse and worse and worse. They put them on antibiotics and they still don't clear it. Antibiotics slow bacteria down so your immune system can get rid of it. So you can see if the person has no immune system, there's nothing there to fight. So the other one is coronaviruses. The coronavirus is a retrovirus as well. And so as COVID has kind of progressed, you start hearing about, okay, well, there's the alpha coronavirus, there's a beta form, there's a delta form. The last one was Omicron. So now they've kind of even stopped trying to give them names. They're just calling them alpha A subtypes, B subtypes. They're all kind of descendant of one original, but they keep mutating. And those mutations affected how infectious it was. So here's like with HIV and AIDS. So the challenge with HIV is to try and develop medications that can block the activity of the virus. So they have what they call antivirals. And this really, HIV really like exploded the research into viruses and medications to block viral function. So prior to this, they really just tried to manage your symptoms. If you had a virus, if you had the flu, if you had a really bad cold, they tried to manage the symptoms. They didn't really have ways of trying to fight viruses. We have antibiotics. Antibiotics only affect bacteria. They do not affect viruses. But in this, this was really the advent of antivirals. And antiviral meds specifically attack certain parts of a viral life stage that human cells don't do. So in this one, notice that RNA becomes DNA. So if you look at HIV, HIV does this. And remember when we were doing protein synthesis, it goes DNA to RNA. It doesn't go the other direction. So that is unique to the virus. The other thing that the virus likes to do is the virus likes to insert itself into the host DNA. So see, like the little blue piece goes in to the gray. So now we have a drug that blocks that. So it blocks the ability of the virus to integrate into the host DNA. Then there are antivirals they developed that are called protease inhibitors. Protease is an enzyme made by the virus that actually damages the cell. And that's what happens just before the virus particles get released. So they develop protease inhibitors to stop that. So they actually tried to attack different steps along the way of a virus's life cycle. But because RNA viruses or retroviruses mutate, they have to change the drugs that they give somebody with this type of infection, change it up so that it decreases any chance of resistance. So resistance would occur if this virus developed the ability to not use the drug that they normally would use, that they normally would block their function. So like HIV could develop a mutation so that the integrase still works. So it could still do that function. So what they have for HIV now, there is about 20 different kinds of drugs. They're all antiviral. Instead of putting you on all 20 of them, they'll put you on like drugs 1, 2, 3. And the drugs 1, 2, 3 block different parts of the HIV life cycle. You take those for 3 months. Then they put you on drugs 4, 5, 6, which block different parts of the virus life cycle. Then they switch it up and they give you 6, 7, and 8. They just keep on changing every 3 months, and that reduces the chance that the virus will mutate and become resistant. That is why now there are people like Magic Johnson who have no detectable virus levels. Their virus levels are so low because they have gone through this antiviral treatment to decrease the number of virus particles so they're not detectable. So now they really say that like they've done lots of studies now and have shown that like you can't transmit it, like even through blood, like through like body fluids and such, that the virus is so low that it's not transmittable anymore. So I know like during the 90s, this was sort of like, seemed like it was like a death sentence for people, but that really led to a lot of research in trying to figure out how do viruses work? How are viruses different than us? And how can we develop meds that target those kinds of activities of a virus? So with COVID, they found that like, so the coronavirus, the one that caused all kinds of havoc in 2020, there's only 30,000 bases, 30,000 nucleotides. That's it. Remember, we're 3 billion? The COVID virus, the SARS-CoV virus, is only 30,000 bases, had only codes for 29 genes. It is really small and very simple. The one thing that they found early on is they found that the protein spike on the surface of this virus, they named it the spike protein, that this protein allows it to bind to a lot of human cells. So if you look, the spike protein, they have it over, they show the little SARS virus here, and there's the little spike protein. So that spike protein binds to what they call an ACE receptor. It's angiotensin converting enzyme, doesn't really matter. But that receptor is found on all of these body cells. So when you look at it, your nasal passages, your lungs, your digestive tract, your heart, your eyes, your liver, your bladder, your kidney, your pancreas, all of these cells have receptors that the spike protein can bind to. Your brain, the placenta. So because it's so common, that's why, I don't know if you remember early on, some people developed a cold. Some people got conjunctivitis. Some people had like GI, they were vomiting and diarrhea for a couple days. There was all kinds of like random, they were like, no, it seemed like nobody was really sure. They were like, I don't know if you have it or not. Because your symptoms and your symptoms are not always the same. Some people would have no symptoms at all, right? They're like, I tested positive, but I'm fine. I don't even have a cold. Like, I didn't even know. I just know that this person got sick, so I tested myself, and I'm positive. But I've never had, I didn't have any symptoms to it. So that is why, is because that protein binds so many different cells. And all the mutations that they've seen, those mutations have actually made that spike be able to bind tighter. So that is where they ran into the question of the variants. So they started off the alpha variant, then the beta, then the gamma, then the delta. But if you look, see how those mutations increased? And with Omicron, it had 32 mutations in the spike protein. And they think that's why it seemed to be more infectious. You didn't get any sicker, but it was more infectious because it was binding better and causing infection. Even if you were exposed to less, you still had a higher chance of infection. The ones now seem about the same. They seem pretty uniform. They haven't had like a big spike in numbers. So in developing these vaccines, Johnson & Johnson, their vaccine is just like the regular flu virus. So they just took a regular cold virus, they inserted the spike protein so that when you got that as an injection, your immune cells got exposed to that spike protein and it helped them make antibodies. And those antibodies also generate or allow for memory cells so that your body now knows what those look like. So every time you get exposed, you end up having more antibodies produced and helps to protect you. The Pfizer and Moderna ones, I really thought they were pretty cool because Pfizer and Moderna, they took RNA. So remember we talked about protein synthesis. So Pfizer and Moderna, they took just RNA, not the entire piece of the spike protein, but just part of the RNA that's used to make that spike protein. They enclosed it in a capsule. So rather than even using a virus, they just enclosed it in almost like a micelle, really, like the substance, like a micelle with the spike protein RNA inside. And that's what you got as an injection. So you didn't have, people tended to have like less cold symptoms, less like side effects from it, but it still triggered antibody production, still triggered immune production. So they have found on average, flu vaccines, people have reactions about 1 per 1.3 million. The COVID vaccines have been less, or about the same, a little bit more maybe, 5 in a million. So it's pretty low numbers. So what they have found now is, this is in the United States because we have totally different healthcare system. I mean, there are some countries that still don't really have any vaccines because of the wealth of the country. With no vaccines, you have a 3% chance of getting it. Okay, that's kind of like getting a cold. It's kind of like getting influenza. It's kind of like, it really depends on what you do. Risks go down if you're working in places of high risk, wearing a mask, because they have found that droplets, sneezes, and droplets carrying through the air is the primary way that infection occurs. Remember they thought originally that maybe if you touch things, like somebody touched the surface, you touch the surface and you get it. They're like, no, you don't even want to know how many times I've wiped these tables down. After every class, spraying and wiping, and then the CDC came out with, that doesn't really work. Doesn't really have a big difference. Droplets are really the big one. The big thing, though, is this. So what's a comorbidity? What does that mean? So if you get it, you have a 3%, or you have a 3% chance of contracting it. Underlying conditions. So what were some of the underlying conditions that they found? So being a diabetic. You know why? Poor circulation, right? Because remember, circulation, blood flow to the extremities. Poor circulation means that immune cells are not going to be able to get to areas where maybe they're needed. So diabetes, what was another one? Lung disease, heart disease. So people that already had lung issues, that just allowed this virus to take up shop. So they were automatically in high risk heart disease. And again, heart disease, if the heart is weakened, then this could put a greater strain on the heart because of fluid in the lungs. So that means that things aren't oxygenated as well as they should be, and that can end up leading to bigger problems. Obesity was a really big problem, but it's still it's hard for people to talk about it because, if you talk about obesity, then you're body shaming people. But obesity, so the more abdominal fat you have, the more that fat pushes up into your thoracic cavity and actually makes your lungs smaller. So now you get an infection in your lungs, and your lungs are already somewhat restricted. And that caused even more problems with trying to ventilate, with that patient trying to move air and having fluids begin to settle in the lungs. So those are the comorbidities. Age, poor circulation, poor immune system response. So those really came out. Children really didn't have a big problem with it. There really weren't very many deaths. In comparison, it was people that were older, people that had weak immune systems, people with these other comorbidities that were a much bigger issue. All right, so that finishes 11. So I will tell you, there is no virus questions on your test. So in this, just go through the mutation information. And I was going to cover the back of this. But the back of this, here's some questions that you should, just down through the mutation part. We covered it in class. So make sure that you cover that. So don't worry about any kind of virus stuff being on this 5th exam. It